Hi, may I come in?
Yes, hello there. Please come in. Have a seat, have a seat.
Excuse me, Professor Binti. I was reading about recombinant DNA technology and I don't understand about this. Can you please explain to me about this?
Further details about it. Okay, so now, before I define to you what is recombinant DNA technology, I'm going to tell you the background of recombinant DNA technology. You've learned about genetics in chapter 5, right? So, we've now got the connection between gene and protein. We know gene encodes protein. We know all that in theory. In fact, people knew this in the middle of 1970s. People were so excited that they understood the idea of how genes give rise to protein through transcription and translation. They read the genetic code that they declared victory. Some said, done with the wonders of life. That was actually the thinking of a lot of the great biologists in the late 1960s. Why did they think such a thing? Well, because they thought they were done with the problem. They thought once you know in principle how the gene gives rise to protein, you could do it. But in practice, nobody could read a single gene. Nobody could even identify a single gene. Wait a second, I said nobody could separate a single gene. Didn't we talk about separating gene material? We can purify the hereditary material away from everything else, but we get all of it together. We don't get individual genes separated from each other. We got the whole mixture of all genes. How are we ever going to separate gene encoding? How are in the world are we going to separate human gene encoding? Okay. So, think about it. That's actually a tough problem, you know? Well, actually there is a brilliant solution that arose in the 1970s to how we could separate the individual genes from each other, but it's not like anybody has seen before. It's called cloning, or the big topic like you said earlier, recombinant DNA technology. So now, what is the definition of recombinant DNA technology? Like I promised to you that I will tell, it is a method in which DNA molecule made in vitro with segments from different sources, or by combining genes from different organisms. Oh wait there, Darin. Oh, my beloved one. Hello? Okay, I'll be in a dash. Alright. Okay, sorry Darin, but I have to go now. We'll talk about tools in recombinant technology tomorrow, okay? Alright Darin, see you tomorrow.
Okay. I'm sorry I'm late. Professor Luigi?
It's okay, Darin. It's okay. You can calm down. Yesterday, Professor Luigi asked me to explain the tools that we are going to use for this DNA recombinant technology. He gonna come today because of his wife. Okay, Darin. There are 5 tools. The first one is target DNA. Okay. It is a sequence of DNA containing gene of interest. Okay, and the second one is restriction enzyme. Restriction enzyme is enzyme which cuts DNA molecules at a restriction site and produce short restriction fragments. Restriction site is the short base pair sequence, and it produces 2 types of cuts, which are sticky end and blunt end. Okay, there are 3 examples of restriction enzyme. The first one is, we call it as ECO-R1. The second one, we call it as SMAR1. And the third one, we call it as BUMH1. Oh my god, oh, what time is it now? I think it's already 5 o'clock. I need to pick up my son. Okay. I think tomorrow Professor Ira is going to explain further tools. He's going to explain further tools. Ram. Oh, yang kita tu. Oh, hi Darin.
Oh, so you are Professor Luigi's students. I'm actually in a hurry, so I can only explain to you 2 of the tools for recombinant DNA. I will explain to you about host cell and modifying enzyme. Host cell is a cell which receives a recombinant DNA and allows the replication of recombinant DNA. Its characteristic is it is able to receive recombinant DNA through transformation process. It is able to maintain the structure of recombinant DNA from one generation to the other, and it is able to amplify the genes produced from the recombinant DNA. Bacteria is the most commonly used as host cell. The reason is because DNA can be easily isolated from or introduced into a bacterium. Bacterial cultures grow quickly, rapidly, replicating any foreign genes they carry. Some of the examples of bacteria that is most commonly used is E. coli. The next one I'm going to explain to you is about the modifying enzyme. Some of the modifying enzymes that is commonly used are DNA ligase and Taq polymerase. DNA ligase is used because it can join the foreign DNA fragments containing gene of interest and bacterial plasmid by catalyzing the formation of phosphodiester bond to form recombinant DNA. Taq polymerase is used in polymerase chain reaction, which is often called PCR. It can replace the DNA polymerase because of the temperature recondition of PCR. It adds nucleotides to the 3' end of a primer in order to produce new DNA strands. Well, that is actually all about host cell and modifying enzyme. Professor Luigi will arrive in just a minute to explain to you about the rest of the recombinant DNA tools. Lastly is about the DNA cloning vector, I think. So, I need to hurry to a class. I'm sorry.
Ram. Oh, yang kita tu.
Sorry Darin, I'm a bit late today because of the hassle that I have at home. Please let me have some time to settle down. Alright? Alright, so where were we? Oh yes, DNA recombinant technology, the tools, right, used in DNA recombinant technology. So the last one is DNA cloning vector, or the meaning vector is transport. So DNA cloning vector is a DNA molecule which can carry foreign DNA or gene of interest fragment into a cell and replicate inside the cell. Alright, got that? Okay, so there are actually characteristics in this DNA vector. It is able to extract foreign DNA in MCS, or as we call it, multiple cloning sites, able to replicate freely in the host cell or a region of replication, initiation, simply ORI. Third, possess selectable genetic marker, resistance to antibiotics, exemplary gracia, ampicillin resistance. Alright, so there are 4 examples that are known until today, which are plasmid, exemplary gracia, PUC 18. It is a DNA molecule that can carry foreign DNA into a host cell and replicate there. And the capacity to carry insertion size is 1 until 10 kilobase. Secondly is bacteriophage, exemplary gracia lambda 2001. It is a virus that infects bacteria, and its capacity to carry insertion size is 8 until 20 kilobase. Third is cosmid, exemplary gracia S cos-1. It is a hybrid vector, meaning combination of plasmid and host gene or phage DNA from lambda bacteriophage, and its cloning capacity is around 35 until 50 kilobase. Now, the last one is YAC, for example, PYAC to be exact. It is a fragment of DNA from the human genome inserted into a yeast cell, and its cloning capacity is the most large of all, which is 100 until 1000 kilobase pair. So now Prof Rosa and Prof Katie and me, Prof B Luigi, has explained to you the 5 tools used in recombinant DNA technology. So are we satisfied for today? Okay, so it's getting late today, so I bet you're tired and hungry, so I'll give you a break until today, and it is our last class for today, and we'll continue our class on steps in gene cloning until tomorrow. Alright.
Thank you, Prof.
Okay, bye, welcome.
Okay, stop.
Prof, yesterday you said you were going to explain to me about the methods in gene cloning.
Oh yes, steps in gene cloning, almost forgot. But today, even better, I'm going to show you the steps in gene cloning. Alright, let's go. So Darin, here's a tube full of plasmid. Inside the tube, there's a circular ring of DNA exists in bacteria. So here is the circular rings of DNA exist in bacteria. Right? It encodes for certain traits that affects how bacteria's appearance, actions, or survival. So let's put this aside. So for example, I would take DNA from a frog and cut it up into pieces. Here is the frog DNA. It encodes certain traits that affect how the frog develops, survives, looks, and now there's a piece of DNA in this genome whose job is to encode certain proteins that the frog uses to survive. So we're going to cut this piece out using a restriction enzyme. You could say restriction enzyme is similar to a pair of scissors. By cutting the DNA at particular parts, as shown by the dotted line, we can get different pieces. As Professor Katie told you yesterday, there are 3 examples of restriction enzyme, and all of them cut in different locations in DNA. The big man we're using to use today is EcoR1. EcoR1 has more than 1 spot to cut, as shown by the dotted lines. So we'll end up with other piece of DNA in addition to our target DNA. Don't worry, Darin, it's okay, we'll deal with the problem later. So cut it off. EcoR1 just doing its job. So there you go, different pieces of frog DNA. Okay, so we're done. And other plasmid too. source of a Prof gene. Alright. So we have new plasmids that we have to stick them into some bacteria. So here are some E. coli with DNA already inside. Note that it will die if we add the ampicillin antibiotics. What the pioneers of gene cloning, who are Cohen and Boyer, did to get the plasmids inside E. coli is to cool the cells down and quickly heated them up, which resulted in little holes in the cell membrane. So here, for example, marked by the black pen marker are the little holes. After we have cooled all of the cells down and quickly heated them up. So, through this hole, the plasmids will slip through and get into the cell. Alright. So, these are the bacteria that we want and didn't want. The one that we want has the Prof gene and didn't want with the wrong piece of DNA or plain plasmid without any Prof DNA, as you can see, even though it is stick by DNA ligase. Alright. Now we put these cells in a plate containing agar. So agar is simply like a jelly with antibiotics. So we put all of this in this plate and leave them overnight for them to grow, and we'll check the next morning. Okay, Darin?
Okay.
So can you please put this in the plate? Okay, thank you. See you tomorrow, alright?
Alright. Hi Darin.
Hi.
Today, our Professor Rosa will replace Professor Luigi in explaining to you what happened to the agar that you guys put in the incubator yesterday. So, let's take it out. As you can see, there are colonies that grew on the agar plate. Now, we need to identify the bacteria that contains our wanted plasmid, aside from these unwanted plasmids that don't have our Prof DNA. The identifying process is called blue-white screening technique. Colonies without Prof DNA are blue because it contains intact leg Z, which produce beta-galactosidase that can hydrolyze X-gal. Colonies with Prof DNA are white because it did not contain intact leg Z, which means it cannot produce beta-galactosidase to hydrolyze X-gal. Okay, so that's all the steps in gene cloning. Do you understand?
Yes.
Alright then, see you tomorrow, Darin.
Bye.
Hello, my name is Professor Kim. You know what? Thanks to DNA recombinant technology, we are able to gain many benefits such as this reduced allergenicity. Recombinant DNA biotechnology also offers the opportunity to decrease or eliminate the allergenic proteins that occur naturally in specific foods. For example, RDNA biotechnology has already been used to dramatically reduce the levels of the major rice allergen. Similar approaches could be attempted with more commonly allergenic foods such as peanuts. So, the next benefits is medical benefits. Recombinant DNA biotechnology brings closer to reality the prospect of commercial production in plants of edible vaccines and therapeutics for preventing and treating animal and human diseases. Possibilities include a wide variety of compounds ranging from vaccine antigens for hepatitis B and norovirus, bacteria, Pseudomonas aeruginosa and Staphylococcus aureus to vaccines against cancer and diabetes. In addition, genetically modified strains of probiotic microorganisms are also possible vehicles for successful delivery of vaccines and digestive aids, such as lactase through the recombinant and the small intestine. Recombinant DNA biotechnology derived vaccines are potentially cheap, convenient to distribute, and simple and safer to administer. So, it can save our people's money. The last one of the benefit is new ingredients. Recombinant RDNA biotechnology can lead to improvements in microorganisms such as bacteria, yeast, and molds that help to convert milk, cereals, vegetables, and meats into a plethora of fermented products, including cheese, cultured meat, sourdough bread, pickles, and sausages. It also can lead to improvements in production of basic ingredients and nutrients such as organic acids, amino acids, vitamins, and enzymes. So that's all from us. Thank you!